brief introduction to Ioannis. I met Ioannis 2-3 years ago in Athens through a common friend from a common department. Ioannis performed his PhD in Patras, the University of Patras, at the biology department, working with Panagiotis Zopretse in Untitled Chapter. And then he moved to Max Planck Institute for Psychiatry in Munich.
I did my PhD there.
You did your diploma in Patras, yes, right. Okay. So under the supervision of Professor Oswald Almeida, and then he had a postdoc fellowship for Riken in Japan, working with Takashima in Alzheimer's disease and stress in Alzheimer's disease. The last 5-6 years, he is working as a researcher in the University of Minho in Portugal, near to Braga. And today he will talk us about his latest findings about the stressed brain as a gauge between depression and Alzheimer's disease. Ioannis?
Thank you, Ioannis. I would like to thank the IMBB for hosting this talk, and Ioannis for the presentation that was here for the rights meeting. But about how chronic stress and glucocorticoid hormones are changing brain plasticity networks related with depression, with the development of depression pathology and Alzheimer's disease. So my talk will be divided in 3 pieces. You will see many times this guy. You're going to see data that I'm not expecting like that all the time. And this is actually the 3 parts we're going to discuss. The first is the impact of chronic stress, chronic psychological stress on Alzheimer's disease and in the initial phase actually. The second part will be more focused on depression pathology and how stress is using the previously known as AD mechanisms to induce, to establish depression pathology. And last part, very fast, we will see a bit about the AD molecules that are more focused on tau outside the central nervous system brain, and a little bit in the peripheral. We're speaking about neurology and stuff that you probably will see here. So, coming back in time very, very fast, it was about more than 100 years ago when the middle-aged young physician at the time, Alois Alzheimer, interviewed the first case, the Augusta D, the first documented, reported case of the disease, asking her name. And she could recall her first name, which was Auguste, but she could not recall neither her last name, neither the husband's name, as well as nothing else. So, indeed, Alois Alzheimer, back in 1907, introduced a new puzzle disease that was later named by Kraepelin, the famous psychiatrist Kraepelin, as Alzheimer's disease. Indeed, he was able, with very, very bad microscopes to our standards, to describe both histopathological hallmarks of the disease. That today we know that these amyloid plaques, which are deposited of peptide, we will see how it's coming, and the neurofibrillary tangles, which is aggregations of cytoskeletal protein tau. So how is the amyloid beta is produced? So there is a bigger transmembrane protein called amyloid precursor protein that it can cleave through 2 different pathways, the non-amyloidogenic, so there is no production of amyloid beta, and the amyloidogenic, which is overactivated in Alzheimer's disease. There is a different cascade, it's cleaved by 2 different cascades, but it's not so important, just keep the fact that this is the main pathway that it is overactivated in Alzheimer's disease. And at the end, the amyloid beta is aggregated and gives these characteristic histopathological plaques in the brain. Now, about tau. So, tau is a basic microtubule associated protein. This is described from this wall on stabilizing, on binding and stabilizing microtubule assembly. It's mainly found in neurons, but also a bit, very little neuroscience, a bit in oligodendrocytes. It's mainly found in action, where more recent evidence when it's stabilized, as I told you. So this binding of tau on the microtubules, it depends on the phosphorylation status. So dephosphorylate tau binds and stabilizes microtubules. Hyperphosphorylate tau, it detaches, and this gives a more dynamic stability. Now, recently we also found that there is dendrites, we and other labs also saw that we can also see tau in the dendrites and inside even the spines, as you will see later also. So what happened in Alzheimer's disease now, what's the problem, is that tau is hyperphosphorylated. It detaches from the microtubules, it accumulates in the soma, and then it drives. And in the same time, we have a destabilized microtubule, which is related with cell death. And as you can see here, more of the tau is in the healthy neuron, where in the disease neuron, we have a translocation at what we call mishort, we will listen more about that, in the dendritics and in the dendritic spines. And this is toxic, we will see how. So in general, the whole field now agrees that in Alzheimer's disease, the main toxic stimuli is amyloid beta. It's this peptide that is overproduced, and in high concentrations is synaptotoxic and neurotoxic. So what amyloid beta is doing is treating a lot of molecules, synapse, neurocircuits, networks. Recently, and we all think that this effect of amyloid beta is mediated through tau, because when we have crossed transgenic animals that overexpress amyloid beta, with tau knockout, we were rescuing the whole phenotype, the molecular behavior, the circuit level. So we know that this is through tau. So different risk factors have been described for Alzheimer's disease, and unfortunately they're increasing. More clinical biological studies add more. So the first, as you know very well, is aging is the prime parameter. But after 60, 65, we have a higher risk for developing the disease. There is also some mutation background in very small populations. So 93 to 95% of 80 patients, they don't have any, at least characterized genetic abnormality or something. Gender or sex is also an important parameter. For example, clinical studies suggest that women have higher risk to develop Alzheimer, as well as environmental parameters such as stress, where our work is more worth it. So what is stress? So basically, it's a physiological mechanism that help in homeostasis. So what is happening when a stress you stimuli? You see snake, or you're stressed because you're late, or I was here coming a bit late this morning, around 11:30. So upon a stressful stimuli arriving, there is an activation of the hypothalamus, pituitary, which is on the kidney, on the top of the kidney, and then we have a release of glucocorticoid hormones that they released, and then they switch off the whole system. Basically, as I told you, this is an adaptive mechanism, but when it is overactivated, we have many studies showing that, for example, overactivation of the system and chronic stress conditions are negatively related with cognitive function in hippocampal. So as much cortical levels you have as you have an atrophy, for example, the hippocampus and the cognitive function go on. Now, on the role of neuronal structure, it is shown by many groups for many years that chronic stress and glucocorticoid hormones, which are the main hormones for stress, are induced neuronal atrophy and synaptic loss related with memory deficits, as well as also with depressive behavior and anxiety. And indeed, depressed patients have higher glucocorticoid levels, and this is one of the clinical parameters to see if your antidepressant treatment is working. You have a reestablishment back to the normal levels of your glucocorticoid levels as a patient. Now, there is also some work showing that many, or a big cohort of Alzheimer's disease does also increase higher glucocorticoid levels, higher stress levels, by the way. And this was suggesting that stress probably was so. When we started many years ago the project, we say, okay, stress is going to induce cognitive deficits, the same with Alzheimer's disease, but they have some neuronal atrophy and synaptic loss. So we think could be an effect. So what we did, we follow a chronic abdominal stress paradigm, which combines different stressors like restraint of the animals. They hate to be restrained. So it is an emotional as well as a physical stressor for them. Or overcrowding, putting them together in a small cage, putting 10 animals in a small narrow cage, that's a stressful environment, or vibrating them. Or we use what is usually used in clinics like synthetic glucocorticoid, dexamethasone. And we use both non-transgenic like amyloid beta infused drug as well as transgenic animals for the Alzheimer. We will select this. So when we stress our animals, what we see, we have an induction of the whole ABP mechanisms. We have more C99, which is the intermediate. We have more production of amyloid beta as well as induction of the enzymes that cut the ABP. I don't go too much detail, so the important thing is that the same happens with tau. We have an increased hyperphosphorylation. We have accumulation in the soma of tau. And we also, when we label it with S-methionine, we see that we have reduced turnover, indicated probably we have reduced degradation as we did later. There were some specific kinases that were activated, which is the main kinases for Alzheimer's disease. It's GSK3 beta, CDK5, and don't stay too much. We use some inhibitors with cells to see how we can block. Some were affected, some there was a sequence. So CDK5 is first, and then GSK3 beta, which we also know in Alzheimer's disease that there's sequential activation of kinases, and they have a cumulative effect on tau hyperphosphorylation. So the same with animals, we have an increased phosphorylation in different epitopes, so it's a wide phenomenon. It's hyperphosphorylation and accumulation. What was interesting is that we see that when we stress your animals for a period of 4 weeks, and then you leave them to recover. So it's normal to recover from stress, all of us will recover, things that we recover. And then when you expose again in a second, the second stimuli, the second stressful stimuli, it is using a higher detrimental effect, both in the molecular and the behavioral effect as you see here. For example, previously stressed animals that they see amyloid beta or glucocorticoid in the second phase, they have higher cognitive deficits we measure by Morris-Water maze. I don't know if you're familiar with the students with this. It's a spatial reference or spatial learning test when we allow the animal to find a hidden platform. So the effect was bigger, and then we use another test which is more for prefrontal cortex cognitive function. It is for reverse learning. So we took the platform. The animals know all the days where's the platform is. You know they learn it because they go down. The stressed animals need more time to find the platform, and this is a clear indication of cognitive deficits. The same with prefrontal. What will happen with prefrontal test? We move from here, that was the platform, all these previous 4 days, we move to the opposite. So we saw the animals just a bit, and then we move it. So what we say to the animals, look, all these days you know that it's here. Now you need to be more flexible and realize that we change it. And we know that stress is also damaging this function by inducing atrophy in the prefrontal cortex. And as you can see, the stressed animals take much more time and did not actually spend more time in the goal. So they were insisting looking here than the control animals that they were did this, what we call behavioral flexibility. This is again a summary of the cumulative effect of stress. So what we see both at the molecular, synaptic, and behavior and pathological level, we see that as much as stress we put, as much that we have increase of tau pathology, we have a very specific distribution starting from entorhinal cortex, hippocampus, and then based on the increasing levels of stress, it distributes a lot. This is actually follows and means what happened in Alzheimer's disease. Because in Alzheimer's disease, the propagation of tau pathology is very specific, starting from entorhinal cortex, going to hippocampus, and then spreading to other cortical areas. And they think that this is happening, and we're now checking with exosome. This is because we have a I would say, kind of primary-like transmission of tau aggregates from one neuron. So the transmission of the neurotoxic tau is between synaptically connected neurons. So the neuron that has that is affected, it's hyperfluid, it aggregates and it throws out. We don't know the mechanism. We think it's exosomes, but we are not actually sure. And then the new neurons pick it up, and then it starts again the procedure. So there's kind of a sitting effect of the new neuron that received the old damaged tau, and it's also damaging its own tau. So as you remember, this is where the risk factors. Indeed, at the next level, we also see that we know that these factors, that the, let's say, precipitators of AD pathology, they interact. For example, AIDS animals are more vulnerable to stress, and AIDS people are also more vulnerable to stress events. On the other hand, women and men, or female and male animals, also respond differently to stress. And so in the next experiment, we use this transgenic animal where we test in 2 different AETs, male and female animals. So we use image stress in order to have, not have a roof effect, in order to have a, let's say, window of opportunity to discriminate, if there is a vulnerability, as the literature says, like women are more vulnerable. And as you can see here, the red is the stressed animals. Female animals of both ages were, damaged. This is again Morris-Water maze, so they take much more time. This is another calculation of the cognitive index. They take much more time and they more effort to reach the platform. Indeed, the old stressed female animals, they never could find the platform. They could not recall. Even after 10 days protocol, usually animals learn very fast. But this is not found in male models of both ages. And this was actually accompanied by the levels of neurotoxic tau aggregates. Because tau is a very soluble protein. It binds and stabilizes or destabilizes microtubules, but it's a very dynamic and natural phenomenon. When it is hyperphosphorylates, it detaches in aggregates. And this is the insoluble aggregate we found, which is where increased by stress only female hippocampus and not in male. And this was accompanied by cell death. So we know that this is neurotoxic aggregates of tau and induces cell death, and we find cell death only in female hippocampus, not in the male. We try to see a bit about how this is happening. So knowing that degradation and aggregation is a balance, especially when a cell is accumulating proteins like tau in our case or synuclein in Parkinson, and we check the chaperone, the molecular chaperones that are doing the work, and we see that they were suppressed in female hippocampus, and we have an induction of apoptotic mechanism together with truncation of tau. This part of tau is also very toxic itself. Besides the aggregates of the big, the small, it's supposed to go to the nucleus and do transcription that is really damaging the cell. And again, this was found only in female hippocampus, not in the male. So again, it's the same mechanism. We continue. So we know that amyloid beta triggers the kinases. We have the hyperphosphorylation of tau, the accumulation, which is reduced degradation, and we have the cognitive deficit. So now we know a bit more about this. That's a schematic representation of what I told you. The transition of hyperphosphorylation to aggregation, it happens through the reduced degradation. This slide, there is a lot of clinical characteristics that are shared between Alzheimer's disease, depression, and anxiety, which actually raise the question if this is common symptomatology are highlighted by common mechanisms. So I highlight this meta-analysis because it's a big study based on more than 100,000 persons in 8 different countries where they clearly state, and then there were other studies that agree on that history of depression increased the risk factor for Alzheimer's disease. And indeed, there are some molecular links between the two diseases where depressed patients, they have amyloid beta levels increased in CSF, and history of depression. So how many times a patient has depressive episodes are correlated via postmortem with increase in the AD histopathology in their brain. So based on our previous work that I present you on the effect of chronic stress on Alzheimer's disease models through the hyperphosphorylation and accumulation of tau, we wonder if this connection is true, then what about if we use depressive pathology using the stress model, but in wild-type animals? And if this is true, if they use the same mechanism, then when we use tau, this is not going to happen. And we just check here is the physiology of the stress, so everything is the same. So when you stress animals, they lose weight in both genotypes, wild-type and tau count. They increase corticosterol levels, very normal response of the HP axis. So everything is working the same at the physiological level with stress. Now let's see what happened with behavior. We check different parameters. We did a lot of different tests on behavior before we go more on the molecular analysis. And as you can see, we have increased mobility in the forced swimming test or tail suspension, which is a clear indication of learned helplessness. This is one parameter of depression. The same with sucrose preference test. It's a test of anhedonia, a hardcore depressive symptom, as Christina would say us many times. So as you can see here, also animals or depressed patients, they don't want to feel the pleasure. They don't go for that. And the same with animals. They drink less sucrose. They prefer more to the water. But what happened with tau knockout under stress? So red is the stress, and this is the tau knockout again. Black is the control and the stress. There was no difference. No difference there. So stress could not induce depressive pathology by any of the different tests in tau knockout. The same with anxiety test, where we have a reduction of the time that the animals stay in the elevated, in the open arms of the here, the elevated plasma test. It is a clear reduction of anxiety. Nothing happened with stress on tau knockout animals. The same with cognition. Also, we have a damage in the Y-mains of the distance or the time the animals spend in the normal land. So what happened here is that we allow the animals to explore these two parts of the maze for 10 minutes, and then 1 hour later we open the third. Usually the animals spend more time or trying to see what is this part that they've never seen. So as you can see, in the stressed animals we have a reduction, which means cognitive deficits. The same with the NOR test. So when we give them another new opposite, I'm not going to details, but all these panels, and I can send you protocols or whatever if you want to, if you're interested more in that. And the same with Morris water test. So the chronic stress induced a delay in the time they find the platform, which indicates cognitive deficits, but there was no stress effect on town locale. Then we did a 3D reconstruction analysis, as you can see here again, as we expected. This is hippocampus, for example. We have a reduction in dendritic atrophy, as expected, by stress in wild-type animals, nothing in town locale. This is the relation. The same with spines. So a reduction of the spine density, the number of the spines of the dendrites, but nothing happened with stress in town locale. This is soil analysis confirming the same, but we have more, less dendritic, less arborization in both hippocampus and prefrontal. And then we check neurotransmitters, both related. We know that we have a reduction of the acetalases, as we found here, and then their turnover by stress in wild-type animals, and this was also blocked by the absence of tau, showing that even, let's say, neuronal probability is not affected. And this is true when we do LTP and as well as so this is for postsynaptic mechanism of synaptic plasticity. I think you are familiar with that. You have key here, and Razin is doing some of the stuff. So stress is going to damage to lower LTP, and this is true in wild type, but this is not in town locale. Even in the presynaptic level, we have a problem, a damage with stress in wild type, nothing town locale, where the overall probability, synaptic probability is not affected. So neurons are there, they function okay, they have just a neuronal atrophy and reduced synaptic connectivity in wild type and not in town locale. Oh, sorry, the MRI, a bit of the MRI. So after working with Dr. Takashima in Tokyo, I like more and more MRI, and now we're trying to do more the network change in the temporal dynamics. So what we found is that we have a reduction of the neuronal activity. This is manganese enhanced MRI in the animals. We have a reduction in the neuronal activity of hippocampus in wild type, but in town locale we have a bit, it is significant, but it's much smaller. So these are saying us again that where we have the damage locally, the overall neural activity of area, and we know that, does not depend only on the local, but it is a part of the network, in which we know that stress is changing the whole overall network. So if one area is activated, it depends not only on the area, but also on the inputs and that receives and sent from other areas. So neural activity is not so straightforward as the dendritic structure or the neurochemistry. Then what we did, we even focused a bit more on the synapses. So we have a very nice detailed protocol for synaptosomal fraction, or to be absolutely correct, is the PSD and RITS fraction, which we have both pre and post-synaptic. So what we find with stress is that when we expose our animals to stress or to glucose, we have an increase of tau hyperstimulation, as I showed you previously, in the cytoplasm, in different, but also we have this tau deducts from the microtubules and also moves to the synapse. So we have the same increase of total tau levels in the synapse. This is what we call tau short, as we said. This is really pathological. It didn't happen in normal. So the amount of tau in the synapse is very low, and it's not usually phosphorylated in the synapse. And so we confirmed that with electron microscopy, and we quantified, so we could see that both in the dendrites and in the synapse, we have an increased tau accumulation and missorting. And in the next step, we also separate, trying to understand what tau is doing there. Because we know if tau is shorted from a de-eclipse field, we know it is synaptotoxic and neurotoxic, but what it's doing there? So what we did in the next level, I separate let's say the PSD from the extrasynaptic. So extrasynaptic is what you see here with blue. It's the extrasynaptic membranes as well as the presynaptic, where pure PSD remains on one side. You see PSD here, synaptophytes in the other. So what we found is that when we have the stress is coming, tau is hyperphosphorylated, mis-shorted inside the synapse, and because it's known that tau binds to fin, and it's a known mechanism for fin, it brings this to the synapse. We have increased fin in the PSD fraction. And this, also because we know that fin is phosphorylated and activates GluN2B receptor, this we also found increased hyperphosphorylation in this specific epitope of GluN2B, which we know again that it initiates cytotoxic signaling, an MDA dependent cytotoxic signal, which is not happening at all in tau knockout animals. So, making it a short, overall scheme of the whole story, as I told you, when stress is coming, we're having glutamate release, which we measure is the same between stress wild type and tau knockout, and we have a lot of calcium entering. Now we're measuring with calcium imaging. So it's very cheap. It's only 400 euros. It's Alcilio from UCLA that suggested they have it even online. So how to do it, how to assemble and do it in animals. So we have in the induction of kinases, they hyperphosphorylate tau detaches from the microtubules, and then this is being shorted to the synapse breaking fin, activated GluN2B, and all the cytosol pathway is activated. But this is not blocked. So it is a tau-dependent mechanism. First, since we see that we have an interest and we know stress is affecting synaptic plasticity, structure, and function, we focus and we do some proteomic analysis together with Mihaela Filipi from Max Planck Institute of Psychiatry, the protocol, when we compare specifically on the synaptosomal fraction of our animals. Again, the same for groups. So what we find very fast, a lot of protein, so nothing basically was changing with stress, very few molecules unrelated with mitochondria in stressed wild type synaptosomal, but this was a big accumulation. You can see the whole long list of proteomic analysis there in stressed tau knockout synapses, which then shows, okay, we checked there is no functional effect on mitochondria. So if so many mitochondrial proteins means that it's not an induction effect or not a transcription level which we check, so it is probably a translocation effect. So what we did originally with TEV analysis in the animals, because we'd like to stay in vivo, is we measure the mitochondria overall, not change among our groups, but as you can see here, no difference between wild type stress and tau, wild type knockout, but there was a significant increase in the postsynaptic localization of mitochondria under stress in tau knockout. And here, just confirming our bldg data, just to see that everything is okay. We have spine loss, and in wild type animals by stress, no synaptic loss in tau knockout. So what we suggest, and now we also have the primary cultural trafficking of the mitochondria, which confirms the same. So what we suggest is that when stress is coming, we have a hyperphosphorylation of tau, and this is damaging the trafficking, the normal trafficking of mitochondria to the spines. And mitochondria has a very beneficial effect for spines. They go there, they absorb calcium, and they provide energy. And this is probably related with the neural atrophy, where in tau knockout, this is happening normal. And indeed, it fits well with the rest of the literature for Alzheimer's disease field, which amyloid beta is doing the same. So when you put amyloid beta in wild type, it damages this trafficking. But if you put it in the tau knockout, there's no problem at all, because probably now we're trying to understand how this is happening with the signaling of mitochondrial trafficking and cargo proteins. These are the papers still. And that's a summary. So I think I provided you with the second part a bit of how stress can impact on signaling, on synaptic structure and function, neuronal structure, as well as the circuit in our brain, referring to both neuronal and to already existing neurons, as well as the newly born neurons, and how this is dependent on tau. So in the last part that I told you, it was probably more interesting for Dr. Garaougias. This has to do with a bit of the tau function outside the central nervous system. And for example, I didn't tell you that in the beginning, not because now it's more recently accepted that Alzheimer's disease is not only memory loss, but we have, for example, pain processing differences. We have also motor deficits in a lot of AD patients. And most of the people, as you know, work on the central nervous system, including us, on Alzheimer's disease. But there is some evidence that also peripheral nervous system is also suffering from tau hyperphosphorylation, and in that case, reduction of total tau levels. So the whole field is saying, if we have, what is happening with tau? How tau is inducing toxicity? Because it's hyperphosphorylated, so it's damaged, it's not working, so it's a loss of normal function, or it's the opposite, because it aggregates, and these aggregates are neurotoxic, so it's a gain of toxic function. So, knowing that tau has a lot of different targets, I'm not going into details, that are related with pain and motor, and we went and checked what happened when there is a loss of tau. So, let's say, trying to answer this question. When you're losing tau, when you're losing normal function of tau, what will happen in PNAS primary afferents, which is the first compartment of pain and motor system? And we focus very specifically on the sciatic nerve, which is a very simple model, trying to understand how this has happened. I'm just saying for people that they're not familiar with it, sciatic nerve has two, and this is when the pain stimuli arrives. We have two fibers, that they're transferring information. It is the A fibers, which is the myelinate for more mechanical and pain, as well as for related with motor function, where we have also the C fibers, which are smaller non-myelinate fibers. So this experimental overview, so we check from all the circuits, starting from the pole, the sciatic nerve, spinal cord, brain, electrophysiology, and structure. We will see that very fast. Tau exhibit reduced pain. This is a bunch of different tests. I don't show all, but as you can see here, as well here, the tau count, they feel less pain. And this indeed was related with the fact that they have less C fibers in the sciatic nerve, but the myelinate fibers were not affected. And when we measure G ratio, it's an index for myelination, and a very clear and unbiased way of measuring myelination in the sciatic nerve, and we found that they have increased G ratio, which means reduced myelination, so hypomyelination for them. And this was also with some molecular and some disturbances in the physiology. But when these animal states, they also have motor deficits. As you can see by different, again, behavioral tests, we have not severe, but we have a mild, we have a, not so severe, but we have a mild, moderate, let's say, motor deficit effect, more behavioral. I'm just okay with that. So what we find with the myelinate, the bigger fibers, which are responsible for the motor transduction and the motor circuit, we find that we have an increased abnormal, so increased degeneration of these fibers in 8 tau count, and the hypomyelination that originally was happening in small fibers, and it was related with pain and the form. By aging, we have also a more severe hypomyelination, but it moves also to the big, the motor-related sciatic nerves. And that actually, we find a bit of the mechanism of how tau and phi interact for the myelination, regulate myelination. This was suggested some years ago in cells, but there was not in vivo evidence about the impact of tau and phi on myelination in the peripheral nervous system. So here we have the proof, the in vivo proof, and here we have some electrophysiology related in vivo electrophysiology, so anesthetized animals, when we measure both muscle and sensory action potentials. For example, if we put lidocaine, we were losing the sensory signal. So we were sure that our peak of the sensorial that we were interested in is here. And as you can see here, we have a reduction, we have an increase by aging, which is normally described, but tau counts, the old tau counts are losing this ability. So it's reflecting also in fitting with both structural and molecular damages that the absence of tau is inducing to both small and big motor-related fibers in the sciatic nerve. That was ex vivo physiology. Again, when I was telling you a summary of the stuff, don't stay too much, just that we have two facts. Decrease of the non-myelinated C fibers by loss of tau, and hypomyelination, which by aging gives damage to the motor deficit. That's why we have the motor damage in these animals. And I think this is the first work that introduced tau and loss of tau in the peripheral nervous system damage. And in a way, the last period, the last 5 years, there was a big discussion since we cannot, all the clinical trials for amyloid beta failed because they have a lot of side effects. And we cannot control hyperphosphorylation of tau because, as I told you, tau phosphorylation is a normal function, it regulates function of tau. If you completely block phosphorylation tau, you're not doing it, you're not dying. It's absolutely problematic. So we cannot regulate tau hyperphosphorylation. But what we can do, there are drugs that there are now that are trying to see where they block aggregation of tau or they reduce tau. But the safety of these drugs, at least in the preclinical studies, are not so. What we are saying that probably tau reduction can have a long-term effect on the peripheral nervous system, on myelination and motor. So we have to be more careful about this suggestion of tau reduction against AD immunization. So trying to summarize, as you know, in the Alzheimer field, there are two schools. It's the people that believe in the amyloid beta, which they call the Baptists, and there are the Taoists, which is obviously believe on tau. So I think at least in our case, and related with stress and depression and the effect of stress, I think we are what we call baptized Taoists, and we do believe, and we have more data about amyloid beta, but there was no time today, that through amyloid beta tau can affect at different levels the molecular synaptic signaling network, because now we're also doing reporting on 3 or 4 different areas the animals at the same time and then doing the behavior. So we can have a real readout when the animal is anxious, what is happening and how it is affected. So our hypothesis started some years ago when I was a PhD student, and I had this stupid idea at the time, which now looks more accepted, that we have an AIDS-dependent decline of neuronal function and increase of atrophy, that depending on the genes and the gender, by aging we have damage of the cognitive status, stress-related, and we have a cumulative effect of stress that can connect and pass from depression and MCI to AD. So if you want to hear more about tau and you're not fully packed with tau in your brain, you can know that it's an outcome webinar that Professor Takashima and I gave last year. So there you can hear it's online, it's free, you can listen, or you can see it, and you can stop it, you can download the video. Or if you still want more about Alzheimer, this is the big Alzheimer forum. You can find a lot about transgenics, about antibodies, about the way you can check things, and this is a big meeting. So at the end, before I stop, I would like to present you my team. Oops, that's not really, it's my team, it's my team, but they're both losers at the end. Portugal here and Braga in Portugal, very bad. Braga got a bit better this year. Anyway, so we have a small team. Sofia, who's finished her PhD. Joana is a PhD student. João is an MD PhD student that now he's doing the 2 years of his PhD in Colombia, doing more with maybe trafficking endosomes and exosomes and RABs. Sarah Hansula, and for sure Nuno Sousa, who is the dean of the school, and actually why I went there is because of him. Our collaborators from Max Planck and Takashima in Tokyo, we're still working with them, my ex-boss, but also Christina from Athens. Ben Volozin, which actually gives us some kind of guidance of how to do stress granules. Without his protocols, we will not do anything. He's a really great guy. Also Clarissa Wells, which is the co-supervisor of João, my student is doing the trafficking, as well as Bruno Brandt. Now we're taking the counseling together. So he sent us one student to train our students in Braga of how to do the trafficking more specifically together with counseling. So if there is any home methods, it's throw away your stress or go to a Gualtar and for a pen, relax when your supervisor say, I need more data. Okay, thank you so much for